So as light passes through the cornea and then the aqueous humor of the anterior chamber and then the pupil and then the aqueous humor of the, I'm saying these words, but as I say these words, you should be going, yeah, got you, until we get out here. So, and then through the aqueous humor of the posterior chamber, then through the lens, then through the vitreous body, and then the light arrives at the back of the eye now, and it passes through here in the back of the eye, what's called the neural retina. And the retina is comprised mainly of three different distinct layers of cells. The first one is a neuronal cell layer called the ganglion cell layer, then bipolar, then the photoreceptive layer. And we finished class last time talking about some of the nuances of rods and cones, and I mentioned genetics to you about color blindness, which is interesting as a way of trying to help you remember the wavelengths for the short, medium, and long cones, to talk about color blindness, keep you interested there. But really today what I want to do, instead of going through all this on-center and off-center field phenomenon and layers of the lateral geniculate, which is all fun, I want to press forward here in the details of the fundamental physiology of vision. So students of physiology need to learn these processes. Now, in my defense, as I walk through this with you, there's certainly organic chemistry and molecular biology here, a lot of it, but that is really not the point here. We just need a taste, is all we need, of the chemistry and the molecular stuff so that we can explain the physiology. So as I go through this, I want you to be aware that when you learn these steps, these 5 steps here, as I talk through them, you have not learned all of it. In fact, as I count them in the Streyer's Biochem text, there are about 21 steps here in doing this process of vision. So the physiology book, this is Miriam's presentation, the physiology book breaks it down to these, and I'm okay with that. All right, now, in order for vision to occur, we are dependent upon a molecule that's located in the membrane of the photoreceptive cells, and that molecule is called rhodopsin. And so rhodopsin really is a combination of a pigmented molecule called a retinal, or sometimes called retinine, that's a chemistry thing, attached to a protein called an opsin. Now, the only difference in this phenomenon in the cone cells, well, let me say this. Why are we doing this in rods? Well, yeah, I mean, our retinas are rods. That's what we are. 120 million of the 125 million cells are rods. And so the only difference really in the process for the cones is that the protein has a different structure. And they're called, instead of calling them opsins, they're called photopsins. So it's a very similar process, obviously. Okay, but this is the molecule of retinal, or retinine, depending on the organic way of describing it, in its isomeric form called the 11-cis isomer. This is the way the molecule exists in the dark. The molecule exists in a bent 11-cis isomeric form. Now, remember from your organic chemistry, when you say 11, you're simply counting the carbons, right? And when you say cis, you mean that the functional groups are on the same side, right? So in this case, we're talking about the place where the bend is made, and so the functional groups are here, these hydrogens. And so whenever the light hits this molecule, it takes on its more stable all-trans form, and you can see what's happened here. This group here has flipped here and is now straightened and formed the all-trans isomer. That is a really important step in vision because the protein will not hang on to this molecule. This is what initiates vision. It is the conversion of this rhodopsin molecule from its 11-cis, more unstable form into its more stable all-trans form. That is what happens to start vision. So as the 11th cis becomes the all-trans written out, then the opsin is released. Now this is a protein. Now notice down here what I've said here. Each of these opsins associated with rhodopsin is associated with more than 100 different G proteins. So the magnitude of what is happening is enormous. When you're talking about millions of cells times hundreds, you're getting a number that you have to use scientific notation to talk about all the molecular biology that's occurring here. Okay, so the opsin, so the light energy causes this transformation to the all-trans. The freed opsin now acts enzymatically to catalyze the activation of a G protein. The G proteins here in the membranes of the photoreceptor cells are called transducins. They're not always called transducins, but you see them referred to this way, specifically in visual physiology. Now, the transducin here, what this says is it catalyzes the activation of phosphodiesterase. But you guys should know, I'm just adding a little bit here and a little bit there, because this is something that should sound familiar to you. If you had to describe the molecular biology of what happens whenever acetylcholine makes the heart slow down, then you already know that this is a general feature in physiology where the G proteins are associated with whatever. In that case, it's actually a channel. And what happens is the G protein is composed of 3 subunits, and normally the alphas separate from the beta-gamma subunits. And if the beta-gamma subunit goes and gets active, it does one thing. If the alpha subunit goes and gets active, it does something else. So this is yet another example of that G protein phenomenon. In this case, it is the alpha subunits that actually bind. Mary doesn't mention that, but I like to bring it up because it helps me remind you of what you've heard about this business before. The alpha subunits bind to the phosphodiesterase. Now, you haven't heard this from me either yet. You haven't heard about the phosphodiesterases. But I did introduce you, just very briefly. We're going to do more and more. I'm going to add more and more to this. When we get to the endocrine system, we're going through it in all of its glory. So, we've mentioned this as we've gone along here. So, I want to remind you that whenever molecules bind to receptors that are in the cell membrane, later in this class, these are going to be hormones. Right now, it's light that starts all this. Whenever molecules bind to receptors in cell membranes, in order to do things inside the cell, in order to make physiology happen, they normally have to activate what are called second messengers. And the big ones that I'm going to emphasize are the ones that are best known. And they include cyclic AMP, cyclic GMP, this one's coming today, diacylglycerol, phosphoinositol bisphosphate, inositol triphosphate, and calcium. The last bits of those are all in the second messenger system, a big picture second messenger system I'm going to do, blah, 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 blah. Okay. So here's the point. Learning what the phosphodiesterase does is meaningful because this is the common enzyme inside cells in your body that stops cyclic AMP and cyclic GMP second messenger systems. Phosphodiesterase is a well-known stop signal, and so you're going to hear about it again. So in this case, the alpha subunit of the transducin binds to the phosphodiesterase. Stop is now the signal. Do y'all see that? I want you to keep seeing this in vision because what we learned about neurophysiology was if you want to make something happen, you need to increase and stimulate. You need to get more positivity in the cell. You need to make it reach threshold. And what happens in vision is actually when my eyes are closed, that's when the stimulation is going on. My eyes are open, the stimulation is stopped as far as the photoreceptor is concerned.
So it's actually a start signal for us in logic that's backwards. So the phosphodiesterase then cleaves this second messenger, and we're not going to walk through how it's made, but there's an enzyme that does it, and you'll learn about adenylate cyclase in the hormone chapter. There's a picture of it at the end of this presentation, so I can say it more than once. Cyclic GMP then is converted by this phosphodiesterase into GMP. It cuts the cycle. It unwinds. It unravels the GMP. And what has been happening in the dark before the light hit this molecule is that there were sodium ion channels in that membrane that were open and held open by this second messenger. Cyclic GMP holds the sodium channels open in the dark. So while it's dark, sodium comes into the cell, and you guys know what that does. That causes excitatory postsynaptic potentials. So when your eyes are closed, there is a flow of sodium into the photoreceptors, which causes a hypopolarization of the photoreceptor. Instead of being at the normal minus 70, the photoreceptor in the dark exists at around minus 40. Now that's bizarre. So I'm going to walk you through this again with pictures out of your textbook so that you can go to your text and look at them also. What happens when light hits is you phosphodiesterase the cyclic GMP, and the sodium channel closes. And when it does, the photoreceptor hyperpolarizes. It becomes more negative as a result of light hitting it. Now, this flow of sodium has a famous term that used to refer to it. It's called the dark current of vision. The dark current. And that's exactly what's going on here. There's a current of positive ions flowing into the photoreceptor. Now, these are two pictures in a row out of your textbook to help you see what's actually happening. Vision is really a double negative, if you think about it. A double negative to make a positive. Okay, let's see if we can work through that. Okay, so this is in the dark. The cyclic GMP-gated channels are open. Okay, cyclic GMP-gated channels. For which ion? Sodium ions, yeah, okay. So allowing a cation influx, I don't know why she just doesn't say that. That's sodium. Cation influx. The photoreceptor is depolarized, or what I said, it is hypopolarized in the dark. In neurophysiology, we'd say that thing is being stimulated. Sodium's going in. Okay? As a result, voltage-gated calcium channels are open in the synaptic terminals, and neurotransmitter is released continuously. Now, this is confusing for students, okay? I understand it's confusing, but you guys need to think. You need to think about the photoreceptors and the bipolar neurons in this system as creating EPSPs and IPSPs and responding to them as though they were action potentials. Because if you stick a little electrode in these guys, in the photoreceptor, you stick a little electrode in the bipolar cell, you will discover they are not making action potentials. That's not what this is. It's this ebb and flow phenomenon of charge, like postsynaptic potentials. That's really what's happening. But the postsynaptic potential is enough here in the dark to cause the release of neurotransmitter from the photoreceptor onto the bipolar cell. Neurotransmitter is released continuously in the dark. Stimulated. This doesn't surprise you, right? That's what we saw at the end of neurons. Calcium comes in, binds to calmodulin, causes the neurotransmitter release. So that makes sense. And now this guy is stimulated all the time. Neurotransmitter is continuously released onto the bipolar cell. Now, if we're going to carry that through, in the dark, we're going to be jamming action potentials into our brain. So look for the click, look for the switch. Okay, the neurotransmitter then causes IPSPs in the bipolar cell. So what kind of neurotransmitter is it that is released here from the photoreceptor? From the photoreceptor cell, it is an inhibitory neurotransmitter. You see that? The stimulation of the photoreceptor in the dark inhibits the function of the bipolar cell. You hear the double negative? Okay. So neurotransmitter then causes IPSP in the bipolar cell, and hyperpolarization results in the bipolar cell. Hyperpolarization then closes the same phenomenon at the end of this guy, acting like a neuron, but these are just postsynaptic potentials, and so the calcium channels inhibiting the neurotransmitter release. So there is inhibition of the neurotransmitter release at the end of the bipolar cell. Now that makes sense. In the dark, I am not stimulating the ganglion cell. Now what question has hit you at this point? What is the question what is the answer? What is the question you want to ask me? See, a good physiology student at this point looks at that and goes, what neurotransmitter? Didn't you want to know that? You learned about all these neurotransmitters, and here we're reading in the text, and it just calls it neurotransmitter. So this is another good example in modern textbooks of things that we don't have worked out yet. Remember the macula densa cells? So there's been proposals of all the famous ones. This glutamate has been proposed in this picture. GABA has been proposed in this picture. Glycine, adenosine diphosphate has been proposed. And so what the textbooks have done, I keep looking for it every year. I don't read the visual physiology literature, but I keep looking for it in the new text to see, okay, is somebody going to find which one this is? Because really, this is a proposed mechanism until you can actually identify the neurotransmitter. Okay, so with all that said, you can see what happens in the dark is no action potentials will occur in the ganglion cell. Where do the action potentials first occur? Do they happen here? No. Do they happen here? No. Do they happen here? This is where they would. But this process in the dark shuts that down. Now in the light, you guys know about the phenomenon, right? What you do is you shut down the sodium channel, and then these guys close, and so the inhibitory neurotransmitter is not released here. So what must be true? So the inhibitory neurotransmitter is not released here. What must be true? What must be true of the nature of the bipolar cell?
Its resting membrane potential is above threshold.
Really, really good, Hannah. Really, really good. It either has to have a resting membrane potential that is above its own threshold. Now remember, it's not making action potentials. So that is a great observation. Or there must be, I'm going to say this because this is exactly what we see in the heart. This is why your heart doesn't need nervous innervation. It does its own thing. There must be enough positive cation channels, passive cation channels, probably for sodium and calcium, in the membrane of the bipolar cell so that it will be excited without stimulation. So we both said the same thing there. These bipolar cells are excitable all the time. And to stop them from exciting the ganglion cell, the photoreceptor cells have to inhibit them. All right, so questions about the process of visual physiology. This is where it really happens, y'all. This is the phenomenon that's occurring in the retina that ultimately results in action potentials being produced in the optic nerve. If you don't make action potentials on the ganglion cells, you don't see. That's vision. These guys right here, these action potentials, that's vision to you and me. Okay, so this is good for you to see. I took a snapshot right out of your book. I put it on the presentation. I recommend, read Miriam's version of this. I'm always recommending you guys read her version and listen to her talk. She's a great A&P teacher. Yes?
Is there any clinical problems with this pathway right here, specifically?
Well, I'm sure. So you mean, is there a specific disease
Yeah.
that impacts the channels of the photoreceptors?
Yeah.
I don't know of one. Wait, it's a good question. So really you're thinking, why would a clinician really care about the excitatory postsynaptic potentials? Yes? It's a fair question. Yeah. Yeah, I don't know. It's a really good question. Okay, so vision is now traveling along the optic nerve. Where is it going? Okay, so visual information is going to several places in the brain, and I'd like for you to know functionally what the importance is of the information arriving at those locations. Okay, so the first one listed over here on the left is a structure in the hypothalamus called the suprachiasmatic nucleus. Now, the neurotransmitters that bring information from the retina into the SCN of the hypothalamus. That is, in the synapses, the optic nerve ganglion cell is synapsing with neurons in the hypothalamus. In those synapses, the neurotransmitter has actually been identified. And there are several of them now called, interestingly enough, they're called melanopsins. Melanopsins. Now, why would you call it a melanopsin? Okay, good. What do you know about the role of the suprachiasmatic nucleus? Biological clock. Okay. And what is the number one most known about rhythm generated and measurable run by the biological clock? Okay, circadian rhythms, good. But what's the number one, the easiest one to measure?
Sleep cycle.
What?
Sleep cycle.
Okay,
Is it the 25 hours?
okay, you're getting there. So I told you guys we have a sleep cycle. I told you we have activity cycles. I told you we have body temperature cycles. But I taught you a specific one by way of connections, a neuroanatomical connection to a gland. What? The pineal gland, which makes a hormone called melatonin. So these neurons that have these melanopsins on them in the SCN are also the cells that are sensitive to the hormone. So this story is getting more and more interesting as time goes on. We know for a fact that this pathway into the SCN is the primary pathway responsible for setting the human biological clock. It's called the retinohypothalamic tract. And so, as you already know, humans have a clock that runs a little slow. The neurons of the SCN run so that we think the days last 25 hours. The reason it doesn't cause physiological problems for us is because the light resets that every day, so that we know about the 24-hour cycle, and that is this pathway from the eyes to the SCN. All right? The second one on this list is a structure called the pretectal nucleus. And this is the one that's a little unfortunate. Its placement on this picture is a little unfortunate because it makes it look like that this is the second place that gets information. So if you follow these blue and yellow lines, you see the pretectal nucleus here. And down here you see the lateral geniculate body of the thalamus. However, if you've had a good anatomy class, then you know already that that is out of sequence. If light is passing into the brain, would it get to the thalamus first, or would it get to, this structure is in the midbrain, as is this one. So which structure would it arrive at next if it's going into the brain? It's passing through the diencephalon, it picks up the SCN, next would be thalamus. And you guys learned in anatomy that the thalamus is the sensory relay station of the human brain, relaying visual information. This is the picture that I use to teach you on that pathway, so you can see that light information passes to the lateral geniculate body of the thalamus on its way to the pretectal nuclei of the midbrain. Okay, so it's a little bit unfortunate that this picture has it. You can see them, they're peeling off axons into the pretectal nucleus here, but they really, this information should come this way. So anyway, I'm not going to belabor that point anymore, other than to say that the pretectal nucleus does receive information from the lateral geniculate body of the thalamus. That's not the only place that gets information from that lateral geniculate body. We also know that in addition to information in the pretectal nucleus, you also get information in the superior colliculus and in the occipital cortex. So what I want to do is give you a functional tag for each of these. Okay? So let's do lateral geniculate pretectal first. This is my functional tag for that one. That information explains that pathway via the optic nerve, the ganglion cells, axons, through the lateral geniculate to the pretectal nucleus, explains why light in one eye causes reflexive constriction in both eyes. And I'd like for you to see this in the lab. I'm going to give you some pen lights next Thursday. I want you to see it for yourself. And this is the pathway from the eyes through the lateral geniculate into the pretectal nucleus of the midbrain. Then, going slightly forward there into a very famous nucleus here called the Edger-Westphal. Edger-Westphal, there's really two ways of describing it. This is the only one you're going to learn from me. The other one goes to the dorsal raphe and the lateral hypothalamus and the spinal cord. That's not the one we're talking about. This is the famous one. This is the pre-cranial nerve 3 nucleus of the midbrain. The most famous. Edger-Westphal is connected to cranial nerve 3. Why do I bring it up? Well, because this is where the crossover happens. The pretectal nuclei of both sides, depending on where the light comes in, will communicate with both. In other words, this pretectal nucleus on this side will talk to the Edger-Westphal on both the right and the left side. Which means that the motor information, the reflexive parasympathetic motor information that is in cranial nerve 3, y'all recognize this one? The ciliary ganglia, that information in cranial nerve 3 is going to go back through the system and impact not just the eye where the light was, but both eyes. So my functional tag for SCN is clock setting. My functional tag for the pretectal nucleus is the pupillary reflex. Okay? Now notice also that information, visual information passes to two other structures here, one called the superior colliculus and the other one called the occipital cortex. Now, I want to do these two, the superior colliculus and the occipital lobe of the cortex, exactly the way the text presents it, so that you can remember it that way. And up here I've told you that the information that goes to the striate or the occipital cortex is referred to as geniculostriate information. Do y'all see that wording? Striate means striped. That refers to the occipital cortex. Geniculo refers to the lateral geniculate body. So the geniculostriate pathway or the geniculostriate system is a system whereby visual information gets to the occipital cortex. It answers the physiology question, what is it? What is it? So this is what we think of. If you ask people, I saw something, this is what they mean. Light hitting the eye, retina, lateral geniculate, occipital cortex, identified. Conscious vision, the geniculostriate. The other visual pathway that includes not only the tectal nucleus, but also the superior colliculus, is a pathway called the tectal pathway. This is a midbrain phenomenon, and I'm going to admit that it includes both, okay? It includes both pretectal and superior colliculus. This pathway, called the tectal pathway, is normally, your book refers to this pathway as a functional component of the superior colliculus of the midbrain. These are the kinds of things that happen with that information. Number one, we use this pathway to do smooth pursuit. That is, tracking an object moving in your visual field. So the microscopic movements that you have to do to keep the image focused on the fovea centralis are done through this pathway. Number two, saccadic movements of the eye. I'm going to ask you to demonstrate this next week in the laboratory with the equipment that we have. I mentioned to you in class last time. I want you to read and watch what your superior colliculus does with your eyes. It is fascinating. So the jerky motions while still, preventing the bleaching of the cone cells of the fovea as you move from word to word, is a function of the tactile system. The pupillary reflex, that shouldn't surprise you, which I just showed you that pathway, includes the ciliary ganglia, and the function of the ciliary body during accommodation. That is, moving the lens so as to accommodate for far and near vision. So really, the tactile system is all of the little nuances that we don't think about with vision. It answers the question, I like this, the tactile system answers the question, where is it? The geniculus striate says, what is it? The tactile system says, where is it? Now, last thing, and I should have started with this, is the visual fields. I pointed this out to you in anatomy, and the picture shows it immediately here. It was the key anatomy observation that led to the Nobel Prize for left and right brain function. And it's this observation that information in the lateral half of each retina is ipsilateral projection, stays on the same side of the brain. And information on the medial side of each retina crosses at the optic chiasm to go to the other side. What that means is everything in the left visual field will project, all this left visual field information will project, as these arrows show, onto the medial side of the left eye in the left visual field. Yes, I said that right. Medial side of the left eye and lateral side of the right eye. So everything out here passes this way and goes to the medial side here and lateral side here, which means everything in your visual field here left is processed by the right brain, and everything, vice versa, everything in the right visual field will be processed on the left brain. So knowing that was the key for Dr. Roger Sperry's work in describing left and right brain function, for which he won a Nobel Prize. All right, that's it for vision. Questions? Okay, the next phenomenon is hearing. I'm also going to give you some tuning forks next Thursday and let you bang some stuff on the desk and test yourself for conduction hearing loss and sensory neural deafness. I'll mention those tests to you here in just one moment. But we got to track sound waves in. So I started with vision by telling you that vision is really about light information. And most of the time when you have a discussion about light, in visual physiology, you're talking about its wave-like properties. But I mentioned to you, you guys have had some physics, you know that light is an interesting phenomenon because it also has a particle nature. Sound, as it turns out, is also a waveform. And sound, as a waveform, has both amplitude and frequency. The amplitude of the sound wave that we hear is what we call loudness, and it's measured in decibels. So when you hear a stadium has a decibel meter, they're measuring how loud the sound is. Our ears are actually not, they are not really designed to deal with loudness and softness as much as they are designed to deal with frequency. So when you have an audiologist test your hearing, they won't have a device where the sound gets louder and softer. Instead, the sounds that you're going to hear are going to be high pitch and low pitch. So this is the way audiologists test it because this is the easiest way to test what's going on. You'll understand that here in just a moment. Sound waves have amplitude and frequency. The amplitude, we say, is measured in decibels. The frequency is measured in cycles per second, and we call that pitch. So as the sound is collected by the auricle, it's channeled through this external auditory meatus in your skull, in the temporal bone, and the sound impinges on a membrane called the tympanic membrane. The sound has a physical phenomenon. It has a mechanical function on that membrane, causing that membrane to physically move. Hearing is dependent on that membrane moving. Now, as it turns out, and you'll see this with the Rhine test for conduction loss, you can also transmit sound by way of bones. You can see the normal physiology is the vibration of the tympanic membrane causes the vibrations of 3 bones in the middle ear cavity. But you can also put a vibrating tuning fork on your mastoid process, and you can hear that. It's not just feeling, you're actually sensing that with your ears. You're hearing it. Then, in the test that you're holding on your bone, just to prove that your tympanic membrane is better at this, when the sound goes away, the vibration has gone away on your mastoid process. If you take the tuning fork and put it in front of your ear, you will still be able to hear it. Because the tympanic membrane is more sensitive to these bones. It's tightly knit to those 3 ossicles in the middle ear cavity, whereas your mastoid process is not. Now that is the conduction test. That's the Rhine test for bone conduction. If you don't hear it when you put it in front of your ear, that means there's something wrong in here with conduction. Yes?
Is that the physiology they use when they do the implant?
Yes, I meant to mention that. Exactly, yeah.
The screws and
Yeah.
everything on it?
That's right. That's the phenomenon, is to put a device on the skull that can make those bones move. Because this is how hearing happens. So that's exactly what they're doing. So the 3 bones are called the hammer, the anvil, and the stirrup, or the malleus, the incus, and the stapes. In order, y'all remember that from anatomy. And the stapes is connected to a structure here embedded in the temporal bone called the inner ear. That bone fits into a little hole in the inner ear, and it is that connection there, the stapes connection. You can see it blown up here in a couple of pictures. That stapes connection there is responsible for delivering the vibration of the tympanic membrane into the inner ear, where hearing is actually going to occur. Now, before we leave the middle ear cavity, I should mention a couple of other things. There's a lot of stuff in here. There's a lot of little blood vessels and nerves that pass through this location. But what should not be here in the middle ear cavity is fluid. It should be air-filled space. If there is fluid in here, it's normally caused by some kind of infection in your respiratory system. Yeah, because this canal, the auditory or eustachian tube, is connected to your nasopharynx, which I pointed out to you in the fall. So this is the pathway by which infectious material gets here. And so there should not be fluid here. And it's dangerous if there is for prolonged periods of time, because fluid here is normally rich fluid in nutrient, and it will stimulate the osteoblasts that are present at these bony connections. And if you have an overproduction of bone, where the bones are connected, they will not move with the same sensitivity they did before. And that is hearing loss. When they won't move like they did before, you were experiencing hearing loss. Now you also have some muscles in here. There's a couple of them, the tensor tympani and the stapedius muscle, that are connected to these bones. And they look to be muscles that are designed to prevent hearing loss with gradually increasing amplitudes of sound. So as the vibrations become intense in the tympanic membrane, these muscles contract in reflex to that, disarticulating the bones from each other, which is why if you're in an environment where the sound is getting gradually louder and louder and louder, you become dulled to that. You walk out of this loud environment that you've been in into a place where it's not as loud, and somebody talks to you, you can't hear what they're saying because your muscles of your middle ear cavity have essentially detached those bones from each other so that they are not causing damage in the inner ear. Does that return? Yeah, it comes back after a while. They relax and the bones hook back together like they're supposed to be, and you can hear again. Okay, now, the place where the stapes is connected is a spot called the oval window. The oval window is connected to the inner ear, which is a structure we call the cochlea. And this picture I showed you in the fall as well. I want to emphasize the frequency phenomenon here because I pointed that out to you. the vibration here causes vibration of a fluid that's found in this inner ear cavity called the scala vestibuli. Now, this is a hard, bony structure that has a soft duct inside it. You remember the anatomy here? The soft duct inside it is called the cochlear duct, and there's fluid, separate fluid in these. This is called perilymph, and that's called endolymph, and slightly different consistency. That's not important. But what I want you to see here is that the vibrations inside this scala vestibuli are caused by the vibrations of the stapes at the oval window. And they will create fluid movement related to the frequency of the sound that the tympanic membrane has picked up. We know, for example, that there is a spread of frequency capabilities of the human ear, some in the neighborhood of between 500 and 20,000 hertz. Now, I had a student here, Ben Talley, a couple of years ago. You guys know Ben? He had a device that would create frequencies, and he had a thing we put on students' ears just so they could see this. And nobody in the lab had anything close to this frequency range. But the textbooks say that this is the typical range, 500 to 20,000. But we didn't have anybody that could pick up frequencies anything close. Like 12,000 was as high as anybody could determine. That's a very, very high pitch. And below 500 is a very, very low pitch. And so I mention these numbers are not to memorize because I have seen firsthand that human ears, at least among the students that were in that lab, could not do this. And mine are horrible. I didn't get anywhere close, especially with the high frequency sounds. He looked at me and said, "There's a lot in this world you're not hearing." Yeah, that's right. So the other phenomenon to notice here is that where these things are, the higher pitches, the higher frequencies, are picked up by the cells of the cochlea, the cells of the organ of Corti cells, closer to the oval window, and the lower frequencies are picked up further out here in the system. So this is a coiled structure. But you can see by looking at this, I hope this is bringing home the point to you, that the ear is really designed for frequency, not amplitude. So when we get weird amplitudes, the system starts shutting down. You get different frequencies, it looks like this whole thing is set up to actually detect different frequencies at different places along this cochlea within the inner ear. Okay, good. Now let's talk about the structure of the cochlear duct that does hearing. It's a structure called the organ of Corti. It has a little membrane on top of it called the tectorial membrane. And then the cells that actually deliver the action potentials to the optic nerve, or the vestibulocochlear, the cochlear portion of the vestibulocochlear nerve, are really modified epithelial cells. This is a weird thing in human physiology, and I always point it out in anatomy too. But the cells that are delivering hearing, and we're going to see them in balance as well, these guys, they do not look like neurons. They don't. They don't look like neurons, but they act like them. So when you bend the little hair cell connections to the tectorial membrane, when the sound vibrations causes this membrane to bend on the cochlear duct, that's what's happening here. So as these guys pass across here, they're causing indentions. They're impressing the fluid down onto that cochlear duct. And as they do, they cause these hair cells off these modified epithelial pieces to bend over. And when they bend over, it opens ion channels in these epithelial cells, stimulates them, and makes action potentials in the cochlear nerve. That is how auditory physiology occurs. The bending of the hair cells of the organ of Corti stimulating the causing action potentials to occur in the cochlear nerve. Modified epithelial cells, very interesting arrangement. Now, the information, the physiology of vision, the physiology of audition or hearing actually occurs in the temporal cortex. So The inferior colliculus does receive some auditory information, and I was tempted this morning as I looked back through this again before coming to class. I was tempted. Y'all know what I was tempted to do?
Make us learn something we don't want to learn.
Okay, that's a student perspective, and I'll take that. Make you learn something you don't want to learn. Okay, we could word that better, though, I think. Yeah?
Throw in something that wasn't originally in your plan to teach us.
Yeah, but like what? Do y'all remember when we went through this in the fall? I didn't just teach you in the fall, I didn't just teach you the general features of the pathways, but we learned the details of them. We learned the details of how each of these things connects. And so I'm going to say now today, remember the inferior colliculus, like its counterpart on top of it, the superior colliculus, is involved in what we might call subconscious hearing things. Like moving the tensor tympani and the stapedius muscles. Nobody thinks to themselves, contract the tensor tympani to protect my eardrum. Yeah, this is an inferior colliculus phenomenon, an auditory reflex phenomenon, in the same way that superior colliculus is move the light across the fovea, move the light on the fovea. That's a superior colliculus function. And so this is auditory, it's a midbrain function here, but we also know that the main conscious hearing location is out in the auditory cortex. I also taught you these crossover pathways in neuroanatomy. I hope one day when you guys take a neuro course, you'll look back and say, Warren taught me that. I can flip through that. Oh yeah, they're making this way too hard. This goes and it's no problem. Okay. Anyway, so a few clinical things here that I want you to test in the lab. The first one, conduction deafness, is where something hampers the sound. You can do this with earwax, a perforated eardrum, what we call osteosclerosis, hardening of the bones. Fluid in the middle ear cavity, yeah. An ear infection could do that, yeah. So after a while, after your third or fourth ear infection, what are they going to do? They're going to put some tubes in the eardrum, and that fluid's going to come out on your pillow, right? So get the fluid out of there. Now, we're going to test this in the lab with some tuning forks. We're going to do that. The lab procedure there is a test for what's called the Rhine bone conduction test. The next one's sensory neural deafness. So if this one's in the middle ear cavity, sensory neural, where's that taking you? Where? Into the inner ear, right. So this is results from damage to the neural structures at any point from the cochlear hair cells to the auditory cortical cells in the temporal cortex. Now there is a test you can do with this too. It's a little more sophisticated. It's called the Weber test. And so I have pictures to show you how to do it and where to put the tuning fork to figure out if there's damage in the left ear or the right ear. Mine shows up beautifully. I got problems in my left ear. Now I knew that already, but the Weber test really demonstrates that for me. Tinnitus is a weird phenomenon where people hear ringing or clicking sounds in the ears in the absence of auditory stimuli. Different people have different ideas about what causes this. I don't buy any of them. It's a phenomenon. I've never had a student tell me that they have tinnitus, but Dr. Webb I'm sure could tell us about a lot of people he's talked to. I keep hearing clicking noises. There is no clicking going on. Meniere syndrome is a famous ear problem. It's a balance issue in which there is some sort of disorder of the labyrinth. What do I mean by labyrinth?
The inner ear.
Yeah. The snail.
Yeah.
The labyrinth is a snail. Disorder that affects the cochlea, of course, that's what we're calling about, and the semicircular canals. So we're about to look at those. Causing vertigo, that's just dizziness. Nausea, that always accompanies dizziness and vomiting. Now what this does, what this Meniere syndrome does for me is it says, okay y'all, there's a connection between hearing and balance. So the last part of this is to talk about balance. And so I go back to the ear picture here and show you the ear structures. But what we want to do now on Monday is we want to talk about this area right here, a little more detail this time. This is called the vestibule. And about these three canals, these are called the semicircular canals. So this area right here with the utricle and saccule, this is the area for static equilibrium and linear movement. This area here, the semicircular canals, this is rotational information. Making circles, rotating your body. And I want you to see the structures and think about how you make action potentials in the nerve. It's the same nerve. It's the same nerve. That's why when there's a balance problem, when there's a hearing problem, they're normally tied together in some way, form, or fashion because they both share cranial nerve 8, the vestibulocochlear. Okay, so I'll do that with you. We'll walk through the physiology together on Monday of the balance structures. Yeah.
What causes hearing loss in elderly
Old age, years of usage.
people? So it just kind of wears out.
Yeah, that's right. So the elasticity in your lens goes away, and you have visual problems. The elasticity in your tympanic membrane goes away, and you have visual problems. The bones in the middle